So it's a pleasure to introduce Shobhna. Shobhna has obtained her BSc from St. Stephen's College, Delhi, MSc from ISC Bangalore. She joined there for an integrated MSc PhD program. But then in her MSc duration itself, she published sufficient number of papers to get a PhD. So she decided to do the PhD elsewhere. So for PhD, she went to Technical University Dortmund. It's pronounced something different in German, but I cannot pronounce it, so I just use the English name. And she was a postdoc in the same city, but in a different institute, Max Planck Institute for Molecular Physiology. In 2016, Shobhna joined us, and how we found her, how she joined, that itself is an interesting story, but we'll not get into that today. And she has, in this short time, established this very, very active group of researchers in the field of molecular biophysics and chemical biology. When we say Shobhna, we think lipids. She works mainly in the area of lipids and lipid-induced signaling, but she's also interested in other research topics. For example, she wants to use chemical biology tools to study cellular signaling pathways. Other than her own research, Shobhna has been instrumental in setting up the departmental cell culture facility. And one thing that I'd like to say also here is that chemists are infamous for building narrow domestic walls. We say that we are organic chemists, inorganic chemists, physical chemists. Even within these, we have very narrow subdivisions. Shobhna is one person who can go beyond these divisions and interact with people. And not only that, even in her teaching, even though for a long, long time she has engrossed mostly in what we would call biology, she has not shied away from teaching quantum chemistry to our first year students. So she is not only a good researcher, she is a very passionate teacher, and she's also very brave. With this, I think that's enough introduction. Now I can hand over to Shobhna.
Thank you, Anindya. Thank you for this very nice introduction. And also thank you, Professor Atre.
Right.
So while preparing this presentation, I realized that I might be facing a very diverse background. So rather than going through all the technical details of my presentation, I decided to mainly focus on three important words: what, why, and how, just to give everyone a flavor of our research program. That being said, as Anindya said, that we work in the field of lipid chemical biology and membrane biophysics, which is my main forte. And we have three different focus areas. For example, development of chemical biology tools, wherein we use chemical synthesis, organic synthesis. We work on membrane biophysics and different applications. And the topic that I will be talking about today is mechanisms of pathogenic lipids in diseases. So with this, as correctly also stated, that we work with lipids. So let me first come to the first question: why lipids and membranes? Because as the title of my talk suggests, that we work with the lipids from infectious agent. So lipids, as you can see, are molecules, biomolecules, which are amphiphilic. That is, they have a polar head group and a hydrophobic chain. And most of us in this audience would associate these lipid molecules to form these phospholipid bilayers. And these phospholipid bilayers in turn form, or are very well known to form, components of the cell membranes in all membranous organisms, be it mammalian cells like ours or bacteria or other agents. However, what I would like to draw your attention is that this phospholipid bilayer is not a very simplified version as shown here. In fact, latest findings have revealed that there is a very heterogeneous cell surface distribution of these lipid domains. And why this particular thing is important is because these different regions or domains of lipids, they basically recruit proteins. As you can see here in this cartoon representation, you have the transmembrane proteins or the peripherally attached proteins. And why they are important is because they actually form the platforms for regulating signaling. For example, for regulating cell division, which we all need for migration, as well as in host pathogen interactions. So the logic that we had was that any agent that can disorganize this kind of lipid domain will have an impact on these cellular processes which are governed by this lateral organization of membranes. To give it or elaborate a little more on that, I have this slide which shows you that the functional roles of lipids are included in signaling. They can affect the activity of proteins that are embedded by interaction with the protein. They can involve in clustering and phosphorylation, eventually leading to immune signaling. And they are actively exploited by pathogens who basically try to hijack these lipid domains in order to enter. Again, coming back to the same argument that any agent that can disorganize these lipids will have implication on the cellular functions. The agent in our case happens to be lipids or pathogenic lipids from Mycobacterium tuberculosis, which causes tuberculosis, so it needs no introduction. But if you see the cell membrane structure, which is very complex, but of interest to us, are lipids which are present in the outermost layer. And for clarity, I show the structures of two main lipids. Let's not go into the nitty-gritty of their names, but the main focus is to understand the molecular mechanism of their interaction with the host cell. So after coming to why of our research program, what basically we do is we basically ask the question, how does these infectious lipids alter the host membrane property? So this is a cartoon representation which tells you how or what we do. So basically we have our mammalian cells and we expose these cells to these purified mycobacterial or lipids originating from bacteria which can cause infection. And we monitor their insertion and ask these main questions that does this insertion or interaction affect organization of the membrane? Does it affect the mechanics? Or does it affect any membrane associated cellular functions? And how basically we address these predominant questions is by using various interdisciplinary techniques. So for example, we use 2-photon microscopy to study membrane organization, wherein we can basically get a surface organization map or lipid domain clustering map of live cell membranes. We use atomic force spectroscopy to study membrane mechanics, and we calculate their properties such as elasticity, stiffness, and rigidity. Then, in order to probe the effect on the underlying actin protein network, which basically provides support to your membrane, we use standard confocal microscopy to study the actin structural rearrangements that is induced by these agents. Then to get a multi-level analysis of membrane fluidity in cells or in vitro, we use different dyes or lipid probes that, as you can see in the schematic representation, can partition at different depths in the membrane. So that enables us to query the membrane fluidity at different regions or depths in the bilayer and how the same are modulated in presence of different infectious components from the bacteria. Then we use infrared spectroscopy, wherein we study the fingerprint regions of different biomolecular vibrations to study biochemical processes, which can be easily done by quantitating their intensity and abundance. Then we use standard cell biology to understand, or cell-based assays to understand the functional implications of what all changes, does the membrane disorganization causes. And for example, what I show here is something known as an autophagy signaling, which is a cell-eating mechanism or a signaling pathway. So we use standard cell biology to query the cellular signaling functions. And last but not the least, we use molecular dynamic simulations in order to map the lipid dynamics and conformation of these lipids. So as I had shown in my previous slide, that these structures are synthetically very intractable. So it's very difficult to modify or study them in isolation. So we, plan to, or we basically adopt these molecular dynamic simulations to get some idea of their structure, so as to correlate it with their functional attributes that we get in our work. So taken together, what we have achieved so far is that these different lipids from this organism are able to insert into the cell membrane of a mammalian cell, wherein it can change the membrane organization. It affects the mechanics, alters the diffusion, it leads to disruption of this actin cytoskeleton, upregulates different kind of biochemical processes, and using other techniques that I have shown, it basically alters the signaling profiles. And what is interesting is that all these lipids, do not have the same behavior, but they actually show a very distinct biological outcome. So what we believe is that maybe this organism can regulate its interaction with this mammalian cell through its various structurally diverse lipids, which cause different biological outcome. And of course, the full scope of implication of this has to be still investigated, and whether we can, let's say, come up with innovative strategies to counter these effects of these lipids so as to curb tubercular infections. So with this, I would like to acknowledge first and foremost my lab members for their hard work and persistence. I would also like to, gratefully thank IIT Bombay for very generous support, not only in terms of finances, but also administrative. And of course, all the institute and departmental facilities that I have used for my work and will continue to do so. My collaborators and all my funding agencies. And finally, I would also like to thank the IIT Bombay Selection Committee for considering my candidature favorably for this award.